Hello. It's been such a long
Hello.
time.
I
Hi.
know, I missed you so much.
Wow.
Me too.
Oh, it's great to be here.
Yeah, great topic today.
Welcome, Mary. Welcome, Victoria. David.
Hello, Victoria. Hello, Mary. And DeWitt.
So the link today has more. So our guest speaker made this link, and in one folder there are all the papers that everyone can download, and then there's the link to the PowerPoint presentation. So
Hello and welcome. Here's our guest.
Hi.
Hello. Will you please tell us how to properly pronounce your name?
Yeah, hi. My name is Yue-Chen Liu.
Nice to meet you.
Yue-Chen.
Yue-Chen. I'm Victoria.
Yeah, thank you. Nice to meet you. Yeah.
Hi, Yue-Chen. I'm
Hi.
Serena.
Hi. Actually, it takes me some time to get familiar with this, how to use this Clubhouse. This is first time I give a presentation on this platform. Yeah.
welcome. Thank you for doing that to come here. We really appreciate it.
Yeah. It's a great opportunity that shall work to a broader audience that's from different field. I'm very glad to have this opportunity. Thank you for the invitation. Yeah.
And this is such an amazing topic. I don't know, I'm from Portugal, so I know it's not on Portuguese people study, but we have kind of also a tradition of seafarers, but
Yeah.
not the first one. So
I also think it's such an interesting topic. I'll be very interested in what you found in terms of the DNA differences and these populations, and yeah, very fascinating.
We're so happy that I will just tell you that we've been already discussing the possibilities of what we might learn from you, because it's an adventurous topic, and it makes you just imagine what people were doing and what it was like, and it's like a really great story is about to unfold. So we're super excited.
Yeah, actually, when we first conduct this study, we do not know that the area that we study in this paper is very, I mean, the population history there is very complex. So it's amazing when we get some results and give our interpretation, and when we talk to our findings to the local communities, and they are also very, surprised by our findings. I hope to talk today will bring more audience to get interested in our work and in our field. Yes.
We will wait a few more minutes. Welcome, everyone. Thank you for coming. And in the meantime, if you want to check out the link, in the link there should be a folder where you can check out all the papers. And then there's also the link for the presentation for today. So, thank you for coming. And in the meantime, I'm sharing the room everywhere. So, we'll start in around 5 minutes.
I'm just looking through the slides now. The slides are gorgeous. It's hard not to just launch into questions.
Yes, slides are gorgeous. Thank you. I hope everyone has access.
Yeah, I also think they're really beautiful. Thank you for making such a wonderful presentation.
Thank you.
How was your day today in general? Good?
Yeah, yes, the weather in Boston today the temperature it became colder, so it's very comfortable day. Yeah, so
Yeah, the
it's great.
same here. I'm so glad the heat wave is over.
Yeah.
For now. I don't know. Is it coming back? I don't know. I don't even want to know.
Yeah.
And I just noticed the acknowledgments. There's so many contributors to this work. It's fascinating.
Yeah, we got actually we got helps from a lot of co-authors and collaborators because they help us collect samples and give us suggestions. And yeah, there are also some local community members that also contribute to our study. So if I have time, maybe I will mention a little about our engagement of the local community in our study. So maybe let's see if I have some time.
We would be happy to make time for that. That part is up to you, how much time you have for us.
Thanks.
We can definitely accommodate that.
Okay.
This is one of the topics that it feels like a loss that we can't pass out popcorn, before. We need that feature in Clubhouse.
We do.
One time I did a room and it was one of my kids' birthday. I kind of mentioned it, and a person in the room sent us cake from a bakery in New York City to our home from somewhere. So that was kind of funny.
That sounds so fun. So we could all send each other
Yeah, very
popcorn.
easy. We can send each other popcorn. I don't know. Is there popcorn on Uber Eats? I don't know.
Well, I often thought how cool it would be in the age of drone delivery if, there's a special class of membership where at the beginning of the event, the drones just show up with snacks.
Oh, yeah, that would be amazing. Finally, a good use of drones, not just bombing people.
Coordinated delivery of snacks for remote events.
Well, do you have the little robot cars driving around where you live? Because when I was in Santa Monica at the outdoor mall, I had to jump out of the way because a little robot, it was the size of a little, like a little wagon that you have when you're a kid, or you pull a kid in like a
Oh,
wagon.
yeah, you have
Yeah,
those. Huh?
yeah, it just sped by, and I had to get out of its way. I felt like I was, the Jetsons.
It was asserting right of way.
Uh, I
Okay.
think we can start. So, welcome everyone to the Science Society here today, and a special welcome to Yue-Chen Liu. I hope I said your name right. And to give everyone a little bit of some information, or to introduce you to the audience a little bit, Dr. Yue-Chen Liu, he's a postdoctoral fellow at the Department of Human Evolutionary Biology at Harvard University. And he got his PhD in zoology at Peking University, China. And there he worked closely with Dr. Yue-Chen Liu and Dr. Stephen O'Brien to investigate the evolutionary history and the natural adaptation of the tiger, Panthera tigris, and other phyllid species using present-day and ancient DNA data. Around 2.5 years ago, he then joined Professor David Reich's lab as a postdoc research fellow, and he began there to use ancient DNA tools to understand the magnificent population history of modern humans around the world. And today we are going to hear a story about the prehistoric migrations into Micronesia, a subregion of Oceania, consisting of thousands of small islands in the northern part of the Pacific, and the matriarchal locality of the early Pacific seafarers revealed by ancient DNA. So this is really an exciting room. I'm very happy that you came here and took the time. And before we start with your presentation, Victoria usually asks a couple of interview questions, if that's okay with you.
Okay, sure.
Yue-Chen, thank you very much, and thank you, Katarina. So the questions are to bring us a little bit into the person behind the research and learn a little bit about you. So what I would like to know is when you can tell us if you can remember, maybe as a child or any time in your life, that you noticed that you had an interest in science, something that first connected you and made you realize that science was something that you maybe not even that you wanted to pursue because maybe it was in childhood, but at any time that was something that you were so interested in.
Yeah, actually, I live in my family live in a place that very near to a zoo. That my parents always take me to the zoo to see the animals and also the plants. So, as you see, as you heard from Katarina, that I have my PhD in zoology. That's why I select the biology and study the animals in my early career. So yeah, I think that's the point that that give me some ideas about what I want to do now and in the future. Yes.
Thank you. That's fascinating. Um, I grew up near an art supply store that I went to every day. And I wondered, and yeah, and so I am an art teacher. And you had this exposure to everything in the zoo and yeah, that's really fascinating. And then what can you bring us along maybe a brief path of events that bring us to your research today, maybe a bit about your studies and interests that led up to this point?
Yeah, actually, at first during my PhD, I focus on the evolution history of animals, especially the felis species like cats and tigers and golden cats from Asia, a small a small body-sized cats. So at that time, I learned some evolution history and also the natural adaptations of this species. And at that time, I found that the collections of the modern samples, collect the species, the specimens from the wild is very hard for the the for us the people who work in the lab and in front of the computer, or for the people who work in the wild. So at that time, we began to collaborate with the museums and zoos, and they help us to collect samples for those from the remains and the furs and the bones that collected in the zoo and also the museums. So that's an aspect of the field of ancient DNA, and that's why after my PhD I come to Professor Rex's lab. The lab is one of the lab who is very at the top levels of ancient DNA field. So that's why I come here to learn and study the the advances in this field, and I hope this will help me to be an independent researcher in the future. Yeah.
Thank you. Well, at this point, you're welcome to deliver your talk, and we can have a Q&A following your talk, or we can have questions along the way that drive your talk along. That's up to you. And sometimes friends in the audience will put questions in the chat, and we can also share those with you so that you can just focus on your talk and your slides if you wish. And so the floor is yours.
Yeah, thanks. And please do feel free to interrupt me if I do not explain something clear, or yeah, if you have any questions, I'm very happy to answer. So yeah, let's begin our today's presentation. Yeah, today I will focus on a story that using our ancient DNA techniques to investigate the popular history of Micronesia, as we mentioned in the introduction from Katerina, that Micronesia is a subregion from the northern part of Pacific, which consists of thousands of small islands that uh, occupied by modern humans around recent 3,000 years ago. So I'm not sure the field that of our audience come from. So I want to start from the materials that we are working on in our lab. So if you come to the second slide of my presentation, so you can see some pictures that the materials that we deal with in our lab. So what is ancient DNA? That ancient DNA is a study of DNA collected from biological specimens that live anywhere from hundreds to hundreds of thousands of years ago. That the ancient DNA is the DNA that collected from specimens, both from the animals or humans or plants that lived in the past. So in our lab, we focus on modern humans. So we collaborated with archaeologists. They help us to collect samples from burials. And ideally, they can see very beautiful burials that have human bones. So we always, or it's ideally, we can collect some bones from cochlea it's there's a figure that shows where the cochlea it is. So yeah, and when we got the cochlea samples, we deal with them in the clean room. We also call it ancient DNA lab. The ancient DNA lab room, that is a room that with positive air pressure, and the people who work in the room that also wear the masks and also gloves, shoes covers, and also the hair nets, and also some disposable lab coats. So all these things, all the protection of the people who work in the lab is not for to protect the humans, but protect the samples. We do not want to introduce contaminates into the room and into the samples that we are dealing with. So after we have the bones, we drill them into a small specimens that generally we extract the DNA from specimens like 50 milligram or more, 50 to 100 milligram in weight. So after we got these small specimens, we drill them into powders, and we wash the powders several times to decrease the level of contaminates in the surface of the powders. So the most struggling issue of the ancient DNA work is the contaminants. So what we want to get is the endogenous DNA from the samples, not the external DNA from either from microbes or from the people who is dealing with the samples. We do not want those contaminants. So we try our best to do several steps to decrease the contamination levels of these samples. So after we do some purification of the bone powders, if you come to the third slides of my presentation, you can see we have how we how we make the bones, how we start from the bones to generate the DNA data, the genetics data, that we extract DNA from the powders after purification, and we constructed the sequencing libraries. And we also, because the ancient DNA fragments is very short and fragmented, it has a very bad quality compared to the DNA that extracted from the cells and the blood and the other materials from the modern humans, for the modern specimens. So a general approach that we increase the coverage of our, or increase the quality of our endogenous DNA, is that we use techniques called DNA SNP capture, that it contains about 1.2 million SNP sites, which the single nuclear polymorphism sites that is designed based on the human reference genome. And we capture those fragments that contains this SNP sites, and then we only sequence those fragments. That will help us to increase the quality of our data. So some of you may have some questions about why we use the cochlea bones but not other bones like teeth or like long bones from our legs or arms. Actually, previous studies that from different labs that have some comparisons between the results of extract DNA from different kinds of bones, and they found that the cochlear bones has the best quality of preserve the ancient DNA. And even in the cochlear bones, the different part of cochlear bones also have different levels of the preservations of the ancient DNA. So the take-home message here is that the cochlear bones always have the best quality of the preservation of the ancient DNA. So the attempts or the efforts that people would like to retrieve DNA from the biological specimens, from the remains or from the burials, it started from the 2010, but only in recent 5 to 7 years, the amount of the ancient DNA data increased a lot. We call it the boom of the ancient DNA data. If you come to the first slide, you can see a figure that display the number of ancient DNA data that generate for recent 10 years. So, but we also find that there is an equal distribution of the generation of the ancient DNA data. You can see the map in the slides. That most of the ancient DNA data is come from the Europe and Asia, or like Siberia and China. But from other continents of the world, like America or Africa or Oceania, there is not many ancient DNA data generated. This is because that Europe and the northern part of the East Asia has a relatively low average temperature, and they have a drier environmental conditions that can help the remains preserve better ancient DNA. But recently, like the study we talk about today, more and more labs try to retrieve the ancient DNA data from other places of the world. So I think in next few years, you will see more studies from other places of the world, like Africa, like America. So the next question comes to here. So after we get the ancient DNA data in hand, what the ancient DNA data can tell us, or generally what an ancient DNA study is focusing about. So if you come to the fifth slide of my presentation, you can see generally three kinds of studies that the ancient DNA field focus on. The first one, in the largest scale, the ancient study can help us to understand the population history through the time. And then the ancient DNA study also can help us to investigate whether there is a local population replacement or population admixture, or whether there is some genetic continuity in a population that's through time. So in a smaller scale, that ancient DNA studies also can tell us if a local community in the past are matrilocal or patrilocal, or the people they're living in the past are polyandry or polygamy. The social structure of the prehistoric people or historic people can also investigate using the ancient DNA technique. So come to the story that I want to share with you today is that we are talking about the region called Oceania. This is a map of Oceania. So based on different criteria, the Oceania is always divided into different parts based on the distance from the islands to the mainland. So the Oceania divided into near Oceania and remote Oceania, but based on different cultural traditions or the different languages spoken in different islands, that the Oceania divided into Melanesia, Polynesia, and Micronesia. But when we talk about the people in history here, there contains two stages of people and events that happen in this area. That the first one happens in around 50,000 years ago. if you come to the 7 slides of my presentation, you can see where the first wave of migration that arrived in Oceania that arrived into. So you can see the first wave of migration happened around 50,000 years ago, arrived in island Southeast Asia, Australia, and also the near Oceania, including the mainland New Guinea, the Bismarck Archipelago, and also the Solomon Islands. But the second wave of migration is happened in a much more recent time. The second one is the one that we are talking about today. We call the second wave of migration into Oceania. Is the Austronesian expansion, which is related to the Austronesian speakers, which is probably originated from Taiwan or the Philippines, that come into the Southwest Pacific region, like Vanuatu, Fiji, and Caledonia. You can see the geographic orientation in the slides in the eighth page of my slides. So they arrive in the Southwest Pacific, like Vanuatu, Fiji, and New Caledonia, and they also arrive in the Polynesia. There is a famous, this is the famous Polynesian triangle that, like Hawaii, Rapa Nui, and the Easter Islands, and also the New Zealand. And also the Austronesia moved to the southwards, to the Pacific. It's arrived in the Micronesia. This is the place that we are talking about today. And due to the recent advances in the archaeology, linguistics, and ancient DNA, also the genetic studies, that the population history in the Southwest Pacific and Polynesia is better known than the northern part of the Pacific. So that's why we conduct this study, and we study and we investigate the public history there. So the first evidence that the evidence of the first human presence in the Pacific, in the Southwest Pacific and Polynesia, is associated with the culture we call associated with the pottery culture we call lapita. You can see the some lapita pottery figures in my slides. So the lapita pottery culture is a red flint pottery that widely spread or found in the near Oceania islands like Bismarck's, and also it was widely found in some remote Oceania islands like Tonga, Samoa, and also in the Southwest Pacific part like Vanuatu, Fiji, and New Caledonia. So our lab began to study the genetics of this Lapita people from several years ago. In 2016, we published a paper that investigates the genetic ancestry of the earliest Lapita people. The samples is collected from Vanuatu and Tonga. The sample is dating to around 3,000 to 2,500 years ago, and our genetic analysis shows that the earliest Lapita people, the people who buried at Lapita sites or immediately post Lapita sites, derived from all their ancestry from East Asian population. They have closest genetic relationship with the modern people who is living in Taiwan and Philippines. So these findings support the out of Taiwan model of the Austronesian expansion, which means the model says that the people Austronesian speakers start from a place, maybe in Taiwan or the Philippines, and spread into the Southwest Pacific and Polynesia, but they do not get mixed with the people who is living in the near Oceania. If you remember that I have mentioned that the first wave of migration into Oceania has happened around 50,000 years ago. That's the people who I'm that's the people who is the local people living in the near Oceania, like New Guinea, Bismarck, or the Solomons. We call these people the Papuans. So we do not see any Papuans ancestry in the early Lapita people. But at the end of the Lapita period, the migrations from the Bismarcks began. They also arrived to the Polynesian. So you can see, it means if we see the genetic ancestry of the earliest Lapita people, they harbored 100% of East Asian ancestry. But if we see the people immediately after the Lapita period, like the Vanuatu people or the Polynesian people after 2,500 years ago, you can see as high as 90% of their ancestry are from Papuans. It means there is a second wave of migration from near Oceania, mainly from the Bismarcks, into the Polynesia and Southwest Pacific region. So if you see all the present-day people in the Southwest Pacific region or Polynesia, almost all of them harbored like at least 25 to 30% Papuan ancestry. It means it is because the second migration from Bismarcks to this region. Well, compared to the Southwest Pacific and Polynesia, there is only a little research or evidence that indicates some scenarios of how people occupy the Micronesia region. Some archaeologists think that the settlements of the Micronesia perhaps began slightly earlier than the Southwest Pacific and Polynesia, because there is another kind of red pottery we call Mariana redwares, which is only found in the Mariana Islands in the Southwest Pacific, in the Northern Pacific. And these potteries are thought to be the first human presence evidence of this region, and this potteries that the earliest dates of this potteries is around 3,500 years ago. So if you remember the Lapita potteries, it's about 3,000 years ago, so it's slightly earlier. Some archaeologists think that the occupation or settlements of the Micronesia is slightly earlier than the Southwest Pacific and Polynesia. But we do not know where this pottery culture come from, and where the people who brought this potteries come from. So the similar ceramics or similar pottery cultures have been found in the Philippines and Sulawesi. So there is still some debates on where the first people of the Micronesia come from. Some people think they have a Philippine origin, but they come from the Philippines or the Taiwan, or some others think that they come from the more southern region of the Indonesia, like Walisa, or some other like Sulawesi or some other place. So this is the first question we want to investigate, or we want to answer using some genetic data in our study, that we want to know where the first people of the Micronesia come from, whether they're from Philippines, or they come from more southern place, more southern part of the Indonesia, more southern place of the Southeast Asia, like Indonesia. So some linguistics also provides some linguistic studies also provide some clues that about the evolution history of the people living in Micronesia. If you come to the 13th page of my slides, you can see a linguistic tree on the left part of my slides. This is the analysis of all the language that include into the Austronesian language family. You can see that all the language spoken in Micronesia that belongs to the Malayo-Polynesian language family, but the language spoken in the Marianas we call Chamorros, and also the language spoken in the Palau we call Palauans belongs to the first split of the Malayo-Polynesian family, which is different from all the other languages that's spoken in the other islands in Micronesia. So other languages from Micronesia, all of them belongs to the Central Eastern Malayo-Polynesian family, language family, except the language spoken in Yarp. All the other Micronesian language belongs to a small clade we call nuclear Micronesian language family. With this language group also contains the Polynesian family and some Polynesian languages and some languages spoken in the Southwest Pacific. So this is the language evidence that sort of consistent with previous hypothesis that the Micronesia and Polynesian that have a very similar genetic history. So, but where is the origin of this nuclear Micronesian language family is also debatable. Or there is several hypotheses about the origin of this language group. If you can see a small map in this slide, there are three hypotheses of the origin of the nuclear Micronesian language. One is from the northern part of the mainland New Guinea. We call the Admiralty Islands. There are small islands in the northern fringe of the mainland New Guinea, and also there is another hypothesis think that this language is originated from the Solomon Islands, and also there is another hypothesis think that this language is originated from Vanuatu. So this hypothesis is also debatable. So another question that we want to answer in our study is that whether this genetic data can help us to trace back where is the Micronesian languages come from, and where is the people of the Micronesia come from. And also, if you see the tree, you can see a different position or different location of the language spoken in western part of the Micronesia, like the Marianas and Palau, and the languages spoken in the Central Eastern Micronesia. So we also want to know whether this tree structures indicate some different history in the western versus the Central Eastern part of Micronesia, whether our genetic data can tell us or can prove our hypothesis. So, and the other thing is that some small islands or some atolls that in the Micronesia that speak the Polynesian languages and also share some Polynesian traits. We call these small atolls like the Nuku'oro or Kapingamarangi. You can see a map in the 14th page of my slide. We call these small islands Polynesian outliers. The questions we want to investigate here is that this is the third question. A third question we want to know is that where this Polynesian culture come from? Can we find some genetic relationship between some specific Polynesian islands and with the Polynesian outliers in the Micronesia? And the fourth question we want to know is in our study, which is one of the most interesting questions that we want to investigate in our research, is that you can see some figures of the Lati pillars in the 15th page of my slides. That the Lati pillars is some stone pillars and with that capstones or cup-shaped capitals that the chamarals used to support their living buildings. You can see different types of lapis stones that shown in the figure. They have different size and different heights and different shapes. But it's interesting that after like 1100 BP, the Lati pillars appeared throughout the Marianas, but we do not see any Lati pillar structures outside of the Marianas. So the question here is that who brought this culture come to the Marianas? Is that the local people who developed this Lati pillar cultures themselves, or some people from other place that take this culture type into the Marianas? That this is the other questions that we want to know in our study. So to answer the above questions, we collected about 134 ancient DNA samples, and also 112 present-day samples across 5 islands in Micronesia, from the western to central part. We have some nice time transect of the samples. Like from Guam, we have the earliest. We have collected samples from the earliest people who is occupied in this island in Guam. It's dating to around 2,800 years ago. And also, in Guam we have collected some Lati period samples who is constructed the Lati pillars and living that period. And we also collect some present-day chamarals from Guam that we can know from this nice time transect. We can know if there is a population turnover or population genetic continuity through the time in these islands. We also have a small time transect from Pompeii in the recent 500 years ago, and we also have collected some present samples from Palau and Truk. And for the Saipan, also the one islands in the northern Mariana Islands, we only have the prehistorical samples who is living in the Lati period. We do not collected the earliest people who is settling Saipan, but based on these samples, we can get a picture of popular history of the people living here. So the first step after we got the data is that we want to know the genetic structures of the sample of the individuals that we collected in our study. If you come to the 17th page, if you come to 17th page, we can see a plot with we call it PCA plot. Each dot in this plot represent an individual. We compared the individuals that we compared newly collected individuals from the Micronesia to all the individuals, all the published data from the individuals in Oceania. So we calculate the PCs, the axis, using the different populations, and then we can see a genetic pattern that can guide us to understand the population structure of Oceania. You can see some dashed lines in this figure, and we actually these dashed lines represent different genetic class, which means the genetic gradients of different people or different group of population. Here is a small example on the right side of the slides. If you can imagine, there are a set of populations who are derived from the same two ancestral groups. One is the 100% Papuan ancestry. The other is 100% East Asian ancestry. But their offsprings have a different percentage of the Papuan ancestry and the East Asian ancestry. So if this is the case in the PCA plot, you can see this population that located in the same lines, but they represent different levels of the mixture proportions. So we investigate, or we found two different clans in Oceania. One class represent the genetic variation in the Southwest Pacific and Polynesia. They connect the Polynesian populations, the Vanuatu populations from Southwest Pacific, and also they connect the Banning speakers from the Bismarck Islands. That represent, we call this clade as the Polynesian clade. And also we also recognize another class which links the mainland New Guinea, the coastal area of New Guinea. The New Guinea groups, the people living in the mainland New Guinea can briefly divide into the Highland New Guinea Highlanders, which is living in the highlands region of the mainland New Guinea, and also the coastal area of New Guinea. So the second clade links the mainland New Guinea Highlanders and the coastal New Guinea people, and also the other people from Micronesia. So from this plot, we know that the people from Polynesia and Southwest Pacific versus the people from Micronesia have different types of Papuan ancestry. One is come from the Bismarcks, and the other is come from mainland New Guinea. And also on the other side of the clade, on the right side of the clade, there is they links a group of green ponds. All of these are from the earliest people who is occupied in the Oceania, like Lapita people from Vanuatu and Tonga, and also the Wunai people, the earliest people that living in the Marianas. So actually, these clades indicates all the populations from Oceania are mixed by different sources of population or different ancestral population. The Polynesian population and Vanuatu, or the other population from Southwest Pacific, are mixed from a Banning speaker, the ancestor of the Banning speaker groups from the Bismarcks, and the first remote Oceania, like the Lapita people. And the Micronesia group are mixed by the mainland New Guinea, the ancestor of the mainland New Guinea, and also the first remote Oceanians, like like Lapita or the Wunai people. So this is our observation in the PCA plot. So then we conduct some statistical methods to confirm our hypothesis. That you can, if you come to the 18 page, you can see we use a statistics method called F-statistics. This is a method developed by our lab. That this method is used to compare if two populations are symmetrically related to one population. So by using this method, you can distinguish the different genetic ancestry within population group. So you can see the people who live in Micronesia and Polynesia are located in different clades, which means they harbor different kinds of Papuan ancestry, and also they have different East Asian ancestry. So the next step, we want to know how many percent of the ancestry. We want to know the admixture percentage or mixture proportions of the genetic ancestry of different population around the Oceania. So we compared the populations from Oceania to the population all over the world. So we found 5 types of different ancestry that widely spread in Oceania. If you come to the 19th page of my slides, you can see we found 2 kinds of East Asian ancestry and also 3 kinds of Papuan ancestry throughout the Oceania region. So in the Micronesia, you can see they harbor 2, they both have the Wunai or Lati Guang related East Asian ancestry and Lapita related ancestry, and they harbor but they only have the mainland New Guinea related Papuan ancestry, but which is different from the scenarios that we observed in the Polynesia and Southwest Pacific. In the Southwest Pacific and Polynesia, they only harbor the Lapita ancestry and another kind of Papuan ancestry, which is combination of the ancestry from the Bismarcks and Salmons and also the mainland New Guinea. So we also, in the next step, after we know the general admixture patterns of this population, we calculate the specific proportions of how many percent of the ancestries come from the Papuan sources and how many percent is from the East Asian sources. So here we come, I think the most interesting point we found here is not the actual percentage of the ancestry, but the things that we found. The East Asian ancestry are mostly come from the females, and the Papuan ancestry is mostly come the males. It means that the mixture between the East Asian ancestry and Papuan ancestry in Micronesia is due to the male expansion, the Papuan males expansion into the Micronesia region. So, but here we cannot know. There's one thing that we cannot know using this approach is that what is the source of the East Asian ancestry in Palau, because we can both use the earliest people who live in Guam, like we call Wunai people in Guam, as the sources of the East Asian ancestry in Palau, but we can also use the Lati people that as the sources to model the present-day people in Palau. So we constructed a more comprehensive model of the evolutionary history of Micronesia. If you come to the 21 page, you can see a tree-like graph we call a mixture graph that it can give us some more details about the evolutionary history of the Micronesia. Here is the most interesting findings here is that we found actually not 2 but 3 lineage of the first remote Oceanians. One is related to Lapita culture, we call this first remote Oceanian Southwest Pacific, and another is called we call it first remote Oceanian Marianas, which is related to the earliest people in Guam. And also, we found a third lineage that we call the first remote Oceanian Palau, which is contributed to the East Asian ancestry in present-day Palau, and also contributed to the East Asian ancestry of the Lati people in Guam. This is the things that we cannot find using previous method, but by using this method, we can distinguish different sources of the genetic ancestry. And we also modeled the admixture time, that when this admixture events happen. So we found that actually there are more than, I think there are 4 types of different admixture happen throughout Oceania. As I mentioned before, if you see the present-day people all around Oceania, except the present-day Chamorros, except the present-day Chamorros, almost all the present-day populations from Oceania have the East Asian ancestry and also have the Papuan ancestry, but they come from different pair of source populations. But you can see here some estimates of the mixture time. They are very similar. Almost all of them comes from the dates is around 2,500 years ago to 1,600 years. So I know this is a large range, but our estimates shows that different mixture events that happen around the similar time. This is a very interesting finding that in our study. This may indicate a radiation expansion or radiation of dispersal of the people living in that period that rapidly occupying different islands in the Oceania. If you remember that I have mentioned there is a question that we want to know is where the Polynesian ancestry come from. We use the same method that we described before to distinguish the Papuan ancestry and the East Asian ancestry. We use this method to investigate where the Polynesian ancestry come from in the Micronesia. We think the Polynesian ancestries come from the Polynesian outliers near the Solomon Islands, like the ancestors from the Tikopia or the Onto Java Islands from the Polynesian outliers. So yeah, and this is the other. If you come to the 24 page of my slides, this is another interesting things that we have found in our study is that we try to study whether or to investigate whether the early Pacific sea fossils are matrilocal, patrilocal. Actually, previous archaeology or anthropology studies tell us that the earliest Pacific islanders are matrilocal. So this is also observed, or this is also supported by our observations in the mitochondrial DNA haplotype of the earliest Pacific islanders. Here, I just compared the Lapita people who is occupied in Vanuatu and Tonga, and and also the Unai people, the earliest people who is occupied Guam, we see different, almost completely different mitochondrial haplotype in Lapita people and Unai people, which means they probably derived from the different ancestral maternal lineage from their ancestral population. So before, yeah, I'm not sure if everyone here is understand what is matriloquality. Sorry, I forgot to explain this, but matriloquality is a role that or a social structure that a woman remains in her mother's household after reaching maturity and brings her husband to living with her family after marriage, and the sons in a family move out of their household after marriage to join their wife's household, which is different from matriloquality in a converse situation. In the prehistoric society structures in Eurasia, most of them or many of them are patriloquality, but in the Oceania, most of the societies or most of the structure is the matriloquality. So here we conducted simulations, mass simulations to see or to investigate the to find the genetic evidence that support the earliest Pacific Islanders and matriloquality. This means, but I after we do these simulations, I do not want to explain the details about simulations, but the simulated results tell us only based on randomly genetic drift, we very hard to see this kind of completely different mitochondrial haplotype in different groups, like we see the E1 and E2 haplogroup in Unai people in Guam, and we see B4 haplotype in the Lapita people. So the simulation results support or tell us that it must be because of some other reasons that make the different make the mitochondrial haplogroups in different existed in different early Pacific Islanders. So so the take-home message here is that our simulation studies or our simulation method is also supported. It suggests that the earliest Pacific Islanders are matriloquality, have a matrilocal structure. So we also, if you come to the 25 page, you can see the burial maps. There are burial maps in Atang beach sites in Guam and Anaguan sites in Saipan. We detected a lot of relatives, the first degree, second degree, or third degree relatives in our samples, which means that many of the individuals that in our studies have a relatively close kinship relationships. So in a summary, if you come to the 26th page, in a summary, we find five streams of migration that helps to establish the people that we see in Micronesia present today. So the first one and the second one is related to East Asian ancestry, and the third one is also related to East Asian ancestry, but is associated with the Lapita pottery culture, which is also existed in the Southwest Pacific and Polynesia. The first migration is associated with the Papuan ancestry, but is different from the ones that existed in the Southwest Pacific Polynesia. The one that exists in Micronesia is related to the mainland New Guinea Highlanders. So, and the fifth one, is the migration related to the Polynesian speakers, probably from the Polynesian outliers near the Solomon Islands. So, yeah, here is the end of my talk, and I want to give special thanks to all the collaborators and also the ancient and present day people who were the source of the samples that we analyzed in this study. And thanks for your time for listening, and I'm very happy to take any questions.
Thank you so much for this wonderful presentation and going over all these data sets with us. For us, it's so fascinating. So thank you so much. And I know that Katie has a lot of questions, so I want to give Katie the opportunity to ask her question. Thank you, Katie, for coming.
Hello, it's Katie. Thank you so much for an amazing presentation. Really encourage everyone to look at the papers and the presentation. And I'm so sorry that I missed the first bit. Thank you so much for being here and welcome to Clubhouse. So, unfortunately, I missed the first bit. And from what I can gather from your paper and also your presentation, that you are collecting samples from a certain part of a body. And I'm just wondering, as someone that works in genetics and genomics myself, however, I'm more of an animal geneticist, I have a lot of trouble with preserved samples. So if something is fixed in formalin or something like that in a museum, I'm wondering if you have had to, and again, I see in your presentation that you usually, excuse my voice, thank you, that you usually collect samples from archaeological sites. I'm just wondering if you've had to deal with museum preserved samples or fixatives at any point. Thank you so much.
Yeah, actually, we always deal with some museum samples. Actually, most of the samples from this study is collected from the Guam Museum, that from the help of our collaborators. The two collaborators, Rosalind and Joanne, they help us to contact the museums and collect the bone samples from museum specimens. But thanks to the preservation, I think they can help us to, in the Guam Museum, of the samples of this study can come from the cochlea part of the bones, that the ones that we think has the best quality of data. So, but sometimes, or sometimes the situation is not so good. Like some of the samples in our lab is come from the teeth, or from the long bones from legs or arms, because you cannot always find the cochlea samples from the barrels or from the museum collections. So it's fortunate that in this study we can get a better quality of samples.
I was just going to say thank you so much for your answer. And I'm sure it's a little bit easier working with vertebrate human bones than elasmobranchs or fish that I work with. Thank you very much. Sorry, Serena, go ahead.
Oh, no. Thank you for that. Yeah, I thought it was interesting that the cochlea was highlighted as a more reliable sample source. But I was really fascinated. It's a wonderful study and it's amazing data. I've always been fascinated in the aspects of what drives a group to leave. And in this study and in these cases, these are vast distances over potentially treacherous waters, where skilled navigation would be required, if not amazing luck. I'm curious. The story that you've painted is that there was all kinds of these events, all kinds of migrations, and it's a complex tapestry of, um, you know, of these radial expansions. But there's different potteries that work in. There's different linguistic aspects in it, and it's an amazingly complex story. I'm wondering if you could comment more on the aspects of what components of either cultural or genetics that drives these expansions, um, and it's just such a curious thing that I mean, was there a tradition of maps and how well known the areas were, or is there a decision where we're going to hit these islands, or um, were there just so many untold failures and few successes? Or I mean, I'm curious. Could you comment more on that if you can?
Yeah, actually, I'm also very curious why, the people living in several thousand years ago who can travel such a long distance from mainland Southeast Asia to these small islands. I talked to this with our collaborators about why, what the drives of the motivation of the people who come to these small islands. Some of our collaborators thinks there might be due to the sources or the resources they live on, uh, like foods or the other sources they need to live in that period of time. But uh, actually, I do not have a very clear answer for a question, but I assume that the living resources is one of the important reasons that let this prehistoric people travel such a long distance to occupy or arrive these small islands. Yeah.
It was also fascinating that this is a predominantly matrilocal structure to the society, and I'm wondering what components would be in play there for that would drive radial expansions. Is it a stable family structure or disagreements with the mother-in-law? Whatever cultural factors might be at play, or is it something much more at a higher level in the culture that out of you mentioned resource scarcity that would drive these expansions, or is it more exploratory? I mean, it's just it's unclear how answerable these questions are, but it's such a fascinating thing to ponder.
Yeah, I agree, but yeah, I think I need to get more materials to see how can I answer this question. But yeah, actually there is some studies that simulate the migration routes of these islanders, that where they use which routes, or use which path to uh, arrive to these islands, or from the Philippines or from Indonesia, which is very interesting. But I think there is not a very conclusive answer about why and what drives what are the drives, or what are the major reasons that motivate these people in that period too.
And lastly, I saw Victoria. I saw you on mic. Just let me get one more in. The technology of the navigation. Is anything known and or preserved to date how accessible these other islands were to navigate, both in terms of seaworthiness and the boats that would be constructed in the we're talking 2300, 2500 years ago,
Yeah,
versus just navigation by the stars, or what other methods was it?
I
Uh,
think
mm-hmm.
Sorry, I'm not sure, but yeah, it's fascinating because the Austronesian speakers not only occupy the Pacific Islands, but they also occupy the other side of the world, like the Madagascar near Africa. So it's also a long, it's also a long journey. So, but I don't know about the navigation stuff. So in that period, I'm sorry about that. Yeah.
Well, thank you. Fascinating work.
Yeah, Yue-Chen, thank you so much. I'm going to somewhat collaborate with Serena, although I hear your answer, but maybe our overall question or the overall message could be to you that you've inspired us so much, and our minds now have so many more questions than we did even after we read the title of your work. So what I was wondering is if there had been any cross-referencing with indigenous wisdom with respect to the migration patterns, and also I'm thinking that the existence of the technology of the crafts that they were using, the boats,
Mm-hmm.
it would that imply that there was more of a cultural basis, and maybe even a genetically not genetically, environmentally, the environmental pressures that would influence that genetic basis for the migration.
Yeah, that's a good question, but I didn't. I mean, um, actually, we do not know the exactly. I mean, the exactly time period of the when this migration happened, but so I do not clearly see the evidence from climate or environment of the uh, of this region, but we know that one of the interesting thing we found in our study is that if you remember that we estimate some admixture events, admixture times from different sources of population, and we found that admixture events that in happened in the Truk and Pompeii, which is in central part of Micronesia, is the time is the similar time have the similar time that the archaeologists find the earliest evidence that people living there. So I'm not sure if there is the mixture events itself is it's a motivate that it's motivate factors that can help people to arrive to a new or unoccupied islands. But yeah, sorry, I'm not. I do not have very much knowledge about the environment conditions in that period. So I'm sorry about that. Yeah.
Oh, please. No, we can only thank you, because just hearing some information that generates a question in our minds is enough. That's the goal, right? We're here because we're passionate about science and scientific thought, and that's what we want to share, and that's what you're sharing with us. So we only have appreciation for you. Thank you so much.
Hi, I have a comment. Um, it's not quite so much a question, but I just think it makes you think. I know some time ago I read stories of the Norse explorers and people like Leif Erikson and so on. And the interesting thing is that in their case, written records. And so you can kind of get a sense of what might, motivate people, like people within a particular community fighting with each other, plus the resources and the methods they use to navigate. So at least in that case, there's some, historical record of it. Just a comment. Anyway, thanks very much for your presentation. I'm sorry I missed quite a bit of it. I'm done.
Thank you. Thank you so much.
Hi, Yue-Chen. Thanks for sharing this wonderful research with Science Society. I have two questions. The one is on the cochlea being the more reliable genetic information source. What among the your colleagues, what are the conjectures? Why is it so, or is there already scientific understanding? My guess is, could it be that because the cochlear structure is much smaller, and comparing to say leg bones, and so that the genetic information is more condensed in a way, and also because the skull bones are somehow relatively better preserved for some reason. That's one question. That's
Yeah, I think the cochlear bones is the best is the best materials of the actually. I think it's interesting that when we compare the because most of our samples come from adult individual, mature individuals, not from a child individuals. But the previous study in our lab that we found that actually for the younger people, for the people from inner child, the teeth would be the best materials that collected ancient DNA. So I think the cochlea, which is relatively good materials that we collect ancient DNA, is because it's it's in the inner part of the bones of the head, so it may not very influenced by the environmental conditions. It will be preserved better endogenous DNA. That's my guess. Uh, but for the child, maybe the teeth uh, is better. It's because it's also in the development stage of a body. So it depends. But for most case of the adult individual, for mature individual, the cochlea is the best. I'm not sure if I answer your question, but yeah, because I do not do much work in the wet lab, but this is my best guess of the cochlea samples, and that's the things I remember from my wet lab colleagues.
sure, and thanks for that sharing. It's interesting. The my second question is in your field of study, I'm curious how the what activities, or what is it like to being a researcher in this field? Do you need to actually physically visiting the field, say to these islands, or
Oh,
how often, or if just curiosity? Yeah.
yeah, actually, the major work of my in this study is the analyst, but I also, but generally, the people like us also visit the field, who we always contact with the archaeologists. We want to keep connection with them because we want to let them know how the best way. I mean, we want to them know the best way to help us to collect the samples, because uh, you the best way to collect samples is when you start to collect them, you need to minimize the contamination level, or you want to let them know which part of the bones is the best one for the ancient DNA studies. The other thing is that we come to the field or come to these islands to do some local community engagement, which means we want to share our results, or we want to share our findings, or both the preliminary ones or the mature ones, to the local communities, that to tell what our interesting findings and get their feedbacks. Actually, in our studies, in this paper or in these studies, we have 2 rounds or 3 rounds of local community engagement in different stage of our our research. The first one is in the preliminary research, we have a very preliminary patterns of our data, and we talk to the local community members, and at that time it's still in the COVID, so so we do this virtually, but a few months ago we go to the Pacific Summit conference, which is a conference happened in Guam. So we go to that place to present our findings to the local community and collect the feedbacks, and to revise our study and some results or descriptions of our findings, and to try our best to let the local community people know what we are doing and what they can know from our studies. Yeah, we are not the ones that are only in the lab, but if we have chance, or if we have some if there is needs, we will go to the field, and we will go to the place that we are studying about.
Well, thank you for that sharing. It's definitely more with the field trips. It's definitely more, better than I would think. Mathematicians just stay shut in a dark room. Thank you.
I have a
Hi.
question, if I may.
Yeah, hi, it's Katie. I was just about to say we've got Nicole, Nabil, Lara, and Kirko. I know that Nabil had an amazing question in the chat too. So Nicole, please go ahead, love. Would love to hear from you.
Thank you. I came to the room very late, so I apologize if you've already addressed this. But were your findings based on the DNA very different from the cultural stories, the anecdotal stories of how people had migrated throughout Polynesia, Micronesia, etc., or did it reinforce what people already thought had happened?
So, if I understand right, your question is, what's the difference of our findings and compared to previous findings, or is that right? Or
yeah, so compared to, say, social, anthropological, cultural information that had been passed down about how people migrated, did the DNA dispute the lore, LORE, the stories, or did it confirm the stories?
okay, yeah. Actually, I think I can take one example of our studies is that previously people think the archaeologists or the anthropologists or linguistics thinks that the Micronesia and Polynesia that were similar with each other, but actually after our genetic research, we found that they are totally different. The only similar thing is that the people living there have both East Asian ancestry and Papuan ancestry, but the sources or the ancestors of this ancestry is different. It's totally different in these two different areas. So that's the one thing that we found differently from previous observations from archaeology and linguistics. The other thing is that previously studies, some of studies think that the people who occupy the Marianas, the first people who occupy Marianas Islands and the Polynesia are the same peoples, but in our study we find that they are different, and also we found a third lineage of people who is occupy the Palau, which is also another lineage of East Asians. But unfortunately, due to our current data, we do not know the exact sources of this population, where they come from, and what the sources of this we call first remote Oceanians. But if in the future we can collect more samples from Southeast Asia islands, so maybe we can investigate this question in a diverse way. Yeah. But most of the findings of our, but some of the findings of our paper is consistent or support previous observation. Not totally turnover, but some are different, and some are the same.
Hi, Yue-Chen. Thank you so much. It was a pleasure to listen to your presentation. Very exciting work that you're doing. I had a question, a clarifying question. I was just wanting to see of your collaborators on this particular project and related projects have been cultural anthropologists.
Sorry, what's the question? I didn't hear clearly. Sorry about that.
No worries.
Can you repeat again?
My question is, how many of your collaborators on this project and related projects have been cultural anthropologists?
Okay, yeah. Actually, take my own study, this study for an example. We have 40 co-authors in our paper, and more than half are the anthropologists and archaeologists who help us to collect samples and help us to review or give our comments about the backgrounds on linguistics or anthropology or archaeologists. Actually, our field is very depends on the collaborations that from the anthropologists and the archaeologists. Without them, we cannot get the samples. We cannot know the questions that we want to investigate using the genetic data. So the collaborations with archaeologists and anthropologists is very important in our field.
Great, thank you for clarifying. I think that's everything for now. Thank you.
Thank you.
Yeah. Hi, Professor. My question is about more on the ancient DNA side. So can you comment at all about what percentages or any different percentages of ancient DNA you found with regards to lineage from the Denisovans and Neanderthal, other human species, and even the lost species, the so-called lost species? Any comment on that? Thanks.
Yeah, actually, the Denisovan and Neanderthal is the archaic humans, which is a close relative to modern humans, but it's totally different lineage. And actually, all the the people in Oceania, like the Papuans and some people in the near Oceania have hovered around 2% or around 2% or so percentage of their ancestry from Denisovans, and like the people from Indonesia also have some percentage of the ancestry from Denisovans, but because of they have a mixture from Papuans, so so we do not estimate the exact percentage of the Denisovan ancestry in our Micronesian samples, but because almost all of them have Papuan mixtures, I think they will have a very little amount of Papuans, a very little amount of Denisovan ancestry introduced by the Papuan mixture. I hope this answers your questions.
Hi, it's Katie. Just wanted to make sure that Kirko and Carl and Golden have a chance to speak as well. But firstly, I just wanted to check in, Professor. I know that we've been speaking for over an hour. I just wanted to check in with your time because I know that I think Dennis as well has another question. So just wanted to check in with your time schedule.
Okay. I think I can answer one more question or so. Yeah.
Yeah, thank you. Who wants to go next? Please flash your microphone, Dennis. Thank you.
Thanks so much for your talk, Dr. Chen. I was looking at the paper and I was curious, when you were doing your purification process, what would you be able to tell us what reagents you used in that process, a little bit more about those methods? I'm always really curious about the methods section.
Oh yeah, you mean the purification of the
Yes.
samples? Yeah. Okay. I think if I remember right, we use because I'm not the ones who is dealing with the wet lab stuff, but if I remember right, this some like alcohols and also some other solutions. You may find the protocols in the reference of our paper. I think we have published the full protocols of how we deal with the ancient DNA samples that in our lab website. You can easily find it. Yeah. Sorry
Excellent.
about that. Yeah. Okay.
No, that's great. I'll definitely look at that. And just quickly, what is I'm trying to wrap my head around the time frame of these observations.
Mm-hmm.
What's the time frame? Is it over a span of 5,000, 10,000, 1,000?
Okay. The time frame of this study is around 3,000 years from the earliest people who occupied Oceania, like 3,000 years ago, and the present day, and to the present day. Yeah. Mm-hmm.
Thank you. Very interesting. And thank you so much for all of your answers and being with us this evening.
Thank you. Thank you. It's my pleasure to do this in this platform and talking to you all. Yeah.
Kirko, did you have a question? I think Kirko is next, right? Please correct me if I'm wrong.
And that's correct.
I don't have any questions right now. I didn't want to ask anything because I kind of missed most of it. And I'm like in the middle of trying to write a SOP. I just wanted to kind of join to see what you guys are talking about. My bad though.
Well, thanks for being here. Kyle, and then Golden and Ed, you had the question?
Yeah, I did. I'm not very smart on this stuff, but he's talking about a specific area. But I'm just wondering, has the DNA been able to trace back to Africa, and did the ice age and when did that happen? That would well, that would yeah, the ice age changed the conditions of uh the migration later on.
Yeah.
I don't.
Actually, sorry. Oh, go ahead. Sorry about my interruption.
Oh, I'm sorry. I just basic questions. I always get confused. We can go back so far, and I'm not sure how accurately we could go back to the uh, Great Migration, or if there was other areas that humans were before the African migration, or it's all. What I hear, it's basically all from Africa, like what 200,000 years ago, when it, going around the land masses. That makes sense. I don't know.
Yeah. Actually, about the DNA from Africa, actually, recent one, I think in this year, or last year, there's some papers about ancient DNA from African began to come out, but the sample size is very small, like 5 or 6 peoples in different area of Africa, because as you know, the African environmental condition, the temperature is very hard for DNA preservation. So, but I think people there is I think at least in my lab, in our lab, there is some projects related to the African area. I hope in the following years you can see more papers from African come out from our lab and also the other lab in the world that focus on the early stage of human evolution. There, because in the African area we focus on different topics. after human evolution. Or for example, like whether they have more admixture from some archaic humans like the Denisova and Neanderthals, which is a little different from the relatively recent stage of the human migration, like my project in recent 3,000 years ago. So it's a different time frame or time scale, but I think more and more studies in that area will come out in the recent
Oh,
in the
thank you.
yeah.
Hello everyone. Can I ask a question?
Sure.
Okay, thank you. I looked at your paper. I don't know how is you extracted the DNA, the method for extraction of the DNA, and how you did the sequencing. Did you use it? Do you use it next generation sequencing or Sanger sequencing in order to build wide genomic data? Thank you.
Yeah. We extract DNA from the bone specimens, like using the ancient DNA extraction method, and we actually with the data is from the next generation sequencing. We extract DNA, construct libraries, and do some capture procedures, which means we just select some fragments of the DNA of the genome that we want to know some specific SNPs in the genome that can help us to investigate the evolutionary history of humans. That yes, it's the next generation sequencing, not the Sanger sequencing.
Okay, thank you. My next question is that if you use the next generation sequencing in order to build wide genomic data, why don't you use it epigenetics in order to know the exact phenotype? Why these phenotypes are common among this specific
The
no,
epigenetics. Okay.
epigenetics, like DNA methylation,
Oh yeah.
ubiquitination, because in the most newest research we see that DNA is not related to most of the diseases, and the sequence of the DNA is not related to the most of the diseases. For example, now it's revealed
Uh,
that many
Colby,
research
I think
revealed that.
we are not talking about diseases, and this goes a little bit away from the topic, and it's really the last couple of minutes we have. So,
Okay,
if you would
thank
like
you,
to say
thank
it on
you.
top,
I
thank you.
Hi, it's Katie. I just wanted to say thank you so much for being here, Professor. Really, really appreciate your time. Thank you, Katerina, and all the Science Society crew for hosting you. Just wanted to, reiterate you're saying that you use SNPs or SNPs, and for anyone that doesn't know, that's single nucleotide polymorphisms that are generated on next generation sequencing platforms. I know some some of us geneticists get into a bit of jargon that people don't understand. So just wanted to reiterate it. And as you said too, everything is in the papers and in the presentation that is pinned at the top of the room. Again, thank you so much for coming to Clubhouse. We really hope we can have you back. I know that we've had so much interest in this room, and I know some people popped in at the end and really want to listen to the replay and excited for it. But I would also love to know, do you have any social media or lab website or anything platforms that we can make sure that we follow along on your journey? Because I'm sure so many of us are so excited to hear about what you're up to in the future. And then I'll turn it back to Katerina after. Thank you.
Thank you.
Yeah. Okay. Thank you so much. For again for did you? I'm sorry. Was I interrupting? Maybe my. I think I have the red bar. Sorry.
I could hear you. Yue-Chen, did you have anything else that you wanted to add? Katie was asking you.
I think yeah, I think you can follow us. You can follow my work and our lab's work in our lab website. You can just search like Rec Lab Harvard University, then you will see our lab website, and you will see we will update our research from different area of the world, not only from the Pacific, but all over the world, from America, Africa, Europe, and other places. So, yeah. And I'm very happy to come here. And if I have more fascinating research findings, I'm very glad to give another, panel discussion here.
Yes, thank you so much. We really appreciate that. And yeah, please, if you, I know a lot of people had more questions, you can reach out to me, and I'll, if I'll try to also answer them or maybe pass them along. And yeah, thank you so much. We thank you so much that you took so much time to answer all of our questions. And yeah, this was an amazing discussion. So we really appreciate it, and we wish you all the funding and all the luck to continue with your research. And yeah, thanks again. And thanks everyone and the audience and everyone to come up here and contribute to the discussion here, asking questions. We really appreciate it. And if you like discussions like these, follow the club. Tomorrow we'll have Dr. Richard Gu talking about artificial microtubules to transport cargoes at 2 p.m. EST. And then on Friday, we will have also at 2 p.m. EST Dr. Bishishia talking about his newest quantum research results and of his colleagues, and how quantum consciousness theories kind of collapse based on the lab results that they have. And yeah, we'll have more rooms next week. And so yeah, thank you so much. And yeah, of course, a special thanks to you, Yue-Chen, and I hear you all back soon, hopefully.
Thank you so much. It's very nice to talk here. Yeah.
Okay, I'll close the room in 3, 2, 1. Bye, everyone.